Welcome you all to this cryo-EM course. Last class we discussed about how to do the negative staining. We also discussed what kind of staining solution do we use. We mainly use uranyl acetate and uranyl formate, how to prepare the uranyl acetate and uranyl formate solution, and what is the basic procedure to store your staining solution. We discussed about all of this. Now then one question it will arise all the time, that okay, if negative staining is good, we are getting a good kind of images, we are getting a good contrast, why we cannot use that negative staining for three-dimensional structure determination? Can we use negative staining technique to determine the three-dimensional structure of any biological macromolecules? Previously, if we just consider 10-15 years ago, we had been using negative staining techniques to determine the structure, and that is called the tilt pair reconstruction, random conical tilt pair reconstruction or orthogonal tilt pair reconstruction. But the major drawbacks, so first I will talk about the what is the advantage of using that negative staining. Negative staining techniques are very easy. It is not that time consuming. If your protein or your biological samples are ready, you can use it immediately if the microscope is available for you, and also it is not very expensive. You can prepare your grade, carbon coated grade in your laboratory, and things will be much cheaper and not that time consuming. And good thing is that you can easily measure the concentration and how homogeneous is your sample and how well distributed. You can easily get those information. But major problem is that whenever you will do the negative staining, you will keep your grid in a dry condition. So what is happening, you are using your grid, you are adding your sample, and then you are blotted your buffer as well as the staining solution. When you blotted excess buffer and staining solution, you cannot immediately insert your grid inside the microscope. You have to wait minimum 15 minutes or half an hour. So grid should be completely dry. When grid is completely dry, you are inserting that holder inside the microscope, and inside the microscope it is under vacuum. So almost 70 to 80% water molecule will be removed from your sample. So your sample will be completely dehydrated. So you can consider that this is a dry sample. And from your biomolecules, if you just remove your water completely, your sample will be collapsed, your sample will be flattened, and that is one of the major problem for the negative staining. When you will do the negative staining, definitely you will get a overall morphology. So if you just consider me as a sample and you want to image these things in microscope, so definitely if you use me as a sample and if you take that image, what you will get? You will get all these details, that overall architecture of my body, the height, head size, diameter of the head, how long is my hand, how long is my leg, you will get all this. But if my body is completely collapsed and if it is flattened, you will not get the proper architecture of all of this. So there will be some kind of changes, even if you will get my head diameter, but if it will collapse, it will be not the exact diameter you will get it. That is one of the major reason we cannot get the exact information, structural information from our negative staining data. And that is the reason this cryo-EM developed, that where we can use at a physiological condition to image our biological sample. This is the main reason. So For negative staining, collapsed and the flattening is a major issue because sample is completely dehydrated. So, we will just go through that same procedure quickly. So, that will help us to go for the next discussion, that okay, how we can use it for other purposes that we already discussed, that we already mentioned that we have to discuss about the carbon sandwich technique also. So, this is our TEM grid again, and if we go, this is our side view of any square, this square from the side, and this is the carbon layer. We are adding the sample. When we have the sample, we blotted the excess sample using this filter paper, and then again we are using this uranyl acetate or format. Then you are using a blotting paper, we remove the excess sample, and then electron beam will pass through that and it will give us an image. So, this is basically the actual procedure how to do the negative stain that we already discussed couple of times. Now, we will discuss about the carbon sandwich method, that where we already discussed that okay, this is the way that our sample will be completely dehydrated. So, this carbon sandwich technique is some kind of technique that is basically helping us not to dehydrate our sample completely. There is a two way that I am going to discuss now. So, previously in 1980 or 1990, this methods was used heavily. Lot of time we are using glycerol or we are using carbohydrate, some kind of carbohydrate solution. So, our sample should not flattened or collapsed. So, what do we have? As I mentioned, for the carbon coating you need a very clean glass slide. If glass slide is not very clean, your that plastic coating or plastic layer will be not detached from the glass slide completely. Here what we have to do, we have to take a mica sheet. That mica we have to use the needle, and if you look at my hand, if you just consider that okay, this is the two mica layer is here. If you look at this one, so this is your mica, and in this mica layer you need a needle, and in between that mica you have to put that needle so you can detach that two mica layer separately. When you detach these two mica layers separately, so inside of that mica we can consider it is a clean, very clean surface. In that clean surface you are adding your sample, and in that sample you can consider that okay, so whatever it is you can mix the glycerol or you can mix the other solution and then make a layer. So, in this particular case what we are doing, first we have a inside part of the mica. Now, you pay attention to this one, that you have a mica here. On this mica, on top of that what we are doing, we are just making some kind of layer on top of that mica, and this layer basically you can use some kind of plastic coating or that form of whatever solution we are using, we are spreading that one, and then we make it completely dry. When we will make it completely dry, this will form a very thin layer which can, if you immerse this entire thing, this plastic layer will float. On top of that what we are doing, we are adding some sample. So, here in this layer we have sample solution also which is dried, and then on top of that there is some carbohydrate or glycerol molecules are there. Now, we are using this mica and coating with a carbon. When there is a carbon layer, carbon will on top of that sample. So, there is a layer of that sample is here on top of that mica, and then there is a carbon coating. And when there is a sample, in between there is a glycerol molecule or carbohydrate molecule. So, carbon cannot pressurize here or push and make the sample completely collapsed. So, in between this sample, based on this with some kind of other molecules like carbohydrate or glycerol, those molecules are there. Now, you take this entire layer and gradually immersed in this water, and in the same way this plastic layer with your sample and carbon coated part, it will start floating on top of this water molecule, top of this water. When it will start floating on top of that water, or you may don't want to use it the water, you just use 2% uranyl acetate or uranyl formate solution here. So, in 2% uranyl acetate, you just try to float your plastic layer on top of this solution. And now, when this plastic layer is floating on top, where you have the sample and then there is a carbon coating, you use a tweezer holding a grid and just pull this entire thing on top of that grid. So, what is happening here? We have the sample with carbohydrate, glycerol, and then top of that there is a carbon layer. The entire thing is floating, and then you are using a grid from the bottom and then pull it up like that. So, then whatever sample you have, it will come and top of this carbon coated grid, then sample, and on top there is another carbon layer. So, then what is happening here? With this grid you have a carbon layer, then you have a sample, and then top there is another carbon layer. So, basically it is forming a sandwich between these two carbon. Your sample is staying, and within this sample you have carbohydrate and glycerol, and that is the reason sample is not collapsed completely. So, sample will be stay as a hydrated sample, and it will keep that almost similar kind of morphology. That is the reason we are using that carbon sandwich techniques also when that cryo-EM is not popular. So, in gradually from 1990, 1995 the cryo-EM became a very popular technique. People can use freeze the sample at physiological condition and image the sample at the physiological condition, and that is the time researchers and scientists move towards the cryo-EM, and this techniques is gradually stopped using by the researcher. But this is a very unique technique that we can use it for sample preparation. There is other methods also that okay. So, here you have a mica, the same way you will detach the mica first. So, when you will detach the mica, inside of that mica we can expect that it is a cleaner and there will be no scratch or no other part which can adhere the sample in a badly. So, you cannot separate the sample. So, inner part of that mica we are adding sample with some other solution that glycerol or carbohydrate, and the sample will absorb on top of this mica. When this sample will adhere here, then we are using the carbon coater and making a carbon film on top of that sample. So, here you can see this is the mica, then we have a sample with other solution, and on top of that we have a carbon layer. Then gradually we will immerse this mica in a negative staining solution, where this mica will separate it from this layer where we have carbon and the sample attached with the carbon film, and from the top you will take the grid again and pull this entire thing. So, in this particular case, if you have a grid here which is holding by a forceps, and when you will pull this grid, what will happen, this grid, top of this grid has a carbon layer. So, when you will do this, you have a grid, when you will take this out, so you have a grid carbon layer, and in top of that one you have sample, and which will coat with a carbon layer. Now, so there is a 2 carbon layer. So, 2 carbon layer here, and then this is the grid. if this is the grid, this is the another carbon layer, and your sample will stay here, and in between there will be a organic solution. It may be glycerol, it may be some kind of carbohydrate which will try to maintain the morphology of our sample. Now we'll go and discuss about that how we can make the metal shadowing for electron microscope. So, metal shadowing for electron microscope is a very popular technique for, if you just consider, is the metal shadowing is mainly used for your bacteria, viruses, any isolated molecules, macromolecules assembly. So, for the getting the high resolution images and mostly getting a better contrast and see the ultrastructure of the biological specimen. So, this bacteria, viruses or isolated any large molecule or macromolecules, those are comparatively big if you just consider DNA. So, what is DNA? DNA, we have a double helix, right? DNA, if we go, it will be just like a double helix. So, you get a minor group, major group, all of these things. So, it is a two strand and it has a phosphate group and then ATGC bases. So, if you compare with a protein structure, protein has alpha helix, beta strand, and they are folded and they are forming a globular structure. In negative stain or TEM, most of the time you can say a globular shape of the protein, but DNA most of the time appear like a thread. So, double stranded, it is, our microscope is not that good enough most of the cases that we can distinguish these two strand properly. So, what it will appear? It will appear like a thread. So, most of the time using normal staining solution, it is bit difficult to visualize this DNA or RNA completely. So, previously metal shadowing we are using for bacteria, viruses or any isolated macromolecules, protein also, but majorly this technique is very popular to visualize the DNA. And how we can do these things? You can follow my slide and you can get each and every details how to do this metal shadowing. I am not going to go line by line, you can follow this procedure how to do the metal shadowing. I will just go to the equipment and describe what is how we can do this, and if you are interested you can read and you can follow this book also. Here what kind of metal we can use it for metal shadow? So, we can use chromium, we can use gold, we can use AUPD, that gold palladium metal, we can use the platinum. Platinum is one of the very common metal, we can use it for that DNA coating. Sometime we can use the chromium, gold also we can use it. So, other metals also we can use it, that whatever it is required, the tungsten, if you want we can use it, but majorly we are using the platinum to coat that DNA. So, here all these details are given, that okay, what is the melting point of all this metal, because we need that kind of temperature to melt the metal, and metal will evaporate and it will distribute evenly on top of the carbon coated grid, and that is the reason melting point is important. And density is also here, based on that we can consider, this is telling that granularity, that okay, what kind of granule it will make when this particular temperature we are using, and if the density of this metal this one, then is it going to give a very fine metal shadow, or if the granularity is very high, if it is not very uniform, the granular whatever it will form, if it is not very homogeneously distributed, it will be not very clean to image. So, based on that we have to select which metal we want to use it. So, here you can see this metal group is giving the very fine distribution, where the platinum is also giving the fine and which is also good enough. So, here what we can do for the platinum we can use the tungsten filament or carbon rod. So, this is a easy to use that when you have a carbon rod. So, carbon evaporation, what we discussed that okay, there is a carbon rod and there is a sharpened carbon rod which touched with each other. So, this is the surface and there is a sharpened carbon rod and there is a friction and which is generating that carbon powder and it will spread all over the grid. So, same thing we can use the similar kind of carbon rod and we can use this metal which is giving us the very fine distribution of the metal to make the coating of that biological macromolecules. So, based on that table we can select. So, mostly we are using the platinum and the carbon rod to coat our sample. So, how we are doing that coating on top of that grid. So, if you look at here, there is a similar kind of equipment we are using. So, most of the time whatever the carbon coater we are using, that similar kind of carbon coater has this facility that we can use it for metal shadowing also or coating our grid with the metal ion. So, lot of time if you just remember that initially I was showing some gold grid. So, that gold grid color if you just remember, it is the gold grid is basically this kind of gold grid we are using. So, lot of time we can use a copper grid and coat with a gold, or you have a carbon coated grid, you have a biological sample on top of that grid and you want to coat with platinum. Most of the time we are using the similar kind of apparatus which we can use for as a metal coater as well as a carbon coater also. So, dual facility will be there. In some cases that dual facility will be not there. Now, if you look at here, so here we have again there is a glass chamber. If you look at here, there is a glass chamber and there is a controller panel. So, all this controller panel what you can do, you can use the voltage, how much voltage you want and how much vacuum is required. The vacuum is very important for all the cases. Why vacuum is so important? Because if vacuum is not good enough within this chamber, even when metal will start spreading, so that there will be a collision between the air molecule and the metal ion and then there will be an uneven distribution of the metal on top of that grid. That is the reason first we have to use a pump system to reach a very high vacuum inside that gas chamber. So, this pump system it will utilize and will reach a proper vacuum inside that gas chamber. When we will reach proper vacuum, then we can start the coating with the metal. During the coating we have a carbon rod. In this carbon rod we have to put this metal wire like this. So, if this is the carbon rod, the metal wire will go like this way all the way to the carbon rod. And now, in this carbon rod when we have this metal wire, it may be the gold, it may be the platinum or it may be the any other metal. Now, when the electric current will go, if you just remember how we are getting the carbon coated grid, there is a voltage, we are using different kind of voltage, we are increasing the voltage. When we are increasing the voltage, it is generating a huge amount heat. When it is generating a huge amount heat, so when it will reach to the melting point of that metal, it will start melting and it will start spreading all over that gas chamber. And if your sample is here at the bottom, and it will just put that metal on top of that. Now, on top of that, if you have a biological sample in this one and metal is spreading in all the way. So, what will happen? Metal will deposit one of this side and this particular other side it will be, there will be no metal coating. So, if this is my biological sample, the metal will coat here in this particular side and the opposite side, if metal will coat this side, opposite side will be empty. So, you can see a drastic contrast difference because of this metal deposition. This side will be black and this side will be white, and you can see this molecule very clearly. So, for the biological science sample, especially for protein or bacteria or viruses, that is not that important, but for the DNA, when there is only a very thin thread is there. So, if there is a deposition and there is a contrast difference between both the side of the DNA will be clearly visible. That is the reason most of the time we are using this metal shadowing for to visualize the DNA. Now, come back to this slide again. So, here this is the internal part of this sample unit, where you can see how it is this carbon rods are there, which will covered with a metal wire, and then your grid or your mica with the sample will stay here, and then this is the unit which will covered with a glass chamber, and this is the rotating sample, this is the plate, it will constantly rotate. So, sample will some metal will distribute uniformly, otherwise metal will not distribute uniformly. So, when this is done, in the next stage what we can do, this mica it will float again. So, there is a carbon coating and then there is a metal coating, your sample on top of that mica, the entire thing it will start floating in the water, and then when this entire layer it will float on top of this water, you take another grid from the bottom of that layer and then pull out and dry it and visualize it in microscope. So, here couple of examples are there. So, this is a DNA and this DNA is looking amazing. Without metal coating it is a bit difficult, and those images are done in a long back, if you just consider almost 30 years ago, and you can see how nicely distributed this entire DNA, and you can see a very long DNA. In some places you can see DNA is denatured also. In here if you see, so there is a two double stranded are connected with each other. Here you can see these two strands are separated from each other. Here there is a strand is broken. So, if you just go to the next one, you can see it much better way that okay. So, this is the entire double stranded viral DNA, where there are some places these two DNA strands are separated, where they are forming the supercoiled and they are packed properly. You can see they are looking like a single stranded here. So, these are all metal shadowing, because if you remember when I showed you the first negative staining sample, I told you that okay, your background will be black and your protein will appear as white, but here you can see because of this metal shadow, the shadow is deposited in such a way, one side it is appearing as white, other side it is appearing as a black metal shadow. Here also you can see a very long DNA strand, and some cases some kind of denature part also you can notice. This is the another DNA molecule, you can see it. So, this is a double stranded PM2 DNA, and also the single stranded DNA, some places it is denatured, and the single stranded DNA you can also see it. So, these are amazing images of the DNA. So, if you do the metal shadow, you will be always get a very high resolution TEM images of the DNA molecule. Nowadays you can also use negative staining, and the microscope quality and lens quality is improved drastically. So, you can use a use this kind of negative staining technique also to image the DNA. Some places your contrast will be not that good. So, that is one of the major problem when you will do the negative staining, because it will be very difficult to distinguish your background from your biological macromolecules. Here you did the metal shadow, that is the reason there is a huge contrast difference between the background and the DNA, and it is easily visible. So, we will today's topic will finish it here, and the next class will discuss about more the good practices and bad practices for negative staining, and what is called the good data and the bad data, as well as application of the negative staining. Thank you all for your attention.